Okay, so welcome to chapter 4 of AP Psychology. It's on sensory and perception. So, firstly, sensory adaptation. Sensory adaptation is a decrease in responsiveness due to constant stimulation. So, if you constantly stimulate it for example, a sound, you'll eventually stop receiving a persistent sound effect. And sensory habituation is when your perception of sensation is partially determined by how used to them you are. For example, you no longer hear traffic from the nearby freeway after having lived in a place for years. So, while sensory adaptation happens in the sensory organs, so this is on your sensory level, sensory habituation happens in the brain. And then cocktail party phenomenon is when if you're imagine that you're talking with a friend and someone across the room says your name or something else of particular interest to you, your attention will probably involuntarily switch across the room. So this is the idea of selective attention. It's humans' ability to change their attention to different things during different times. So there are 5 senses that we need to cover in this chapter. The first one is vision. So the first step of vision is when you gather light. So light is reflected and gathered by the eye. The color depends on light intensity. Light intensity and light waves. And once the light is gathered, you go through what's going to happen within your eye. So the light enters the cornea. The cornea helps you focus and protects your eye. So your cornea is right here. And the pupil controlled by eye basically controls the light and it dilates when there's a lot of light. Okay, and then within your eye, you also have your lenses. And then when passing through the lens, image is flipped upside down and inverted. Which means that this goes through your retina. Your retina is basically this screen at the back of your eye, and it has special cells, special neurons that you're going to activate. So this goes through, which is right here. So basically, your cornea goes through your iris goes through your retina flips the image so that you see it the right way and not upside down. And after that, you go through transduction. Transduction means that your stimulus becomes your neural signals. There are several layers of neurons on your retina. So all of these are all neurons. And you have 2 types of neurons, cones and rods. So I'm going to write it right here. You have cones, have rods. So cones are activated by color. So just remember C and C, cones color. And you have rods that respond to only black and white. So you very center of your retina, you have indentation, which is right here. So the indentation is what we call fovea. Fovea has the highest concentration of cones. If enough cones and rods fire, they activate bipolar cells. If enough bipolar cells fire, you get ganglion cells. So continuing on the process. cones and rods activates bipolar, which activates ganglion. And then your optic nerve sends impulses to lateral geniculate nucleus, which is called LGN. And this is within your thalamus. And if you recall from the other chapter, thalamus is where is the flight tower. So everything goes there and gets retransmitted to something else. So all your visual cord, all your neurons, neurotransmitters go through your thalamus, and then it gets sent to your visual cortex. So the blind spot that you have, which is the term right here, is when you don't have any rods or cones. So your optic nerve has impulses from the left retina, which is the left hemisphere, and your right retina, which is your right hemisphere. And the spot where it crosses is called the optic chiasma. Okay, so moving on to brain. Your brain kind of takes in the visual cortex, and impulses are activated by feature detectors. There are different theories of color vision. Firstly, you have trichromatic theory. Trichromatic theory means that you have 3 types of cones in your retina: blue, red, and green. However, this theory is not a really accepted theory because it does not explain after images, and we'll go to after images afterwards. And there are 2 types of colorblind. You have dichromatic colorblind and monochromatic colorblind. For monochromatic, you only see shades of gray. And for dichromatic, in pairs. So red and green is a pair, and blue and yellow is a pair. So you can, if you don't see red, you don't see green. If you don't see blue, you don't see yellow. So they all come in pairs. And then you have the opponent process theory, which basically says that the sensory receptors in your retina come in pairs. So red and green, blue and yellow, black and white. So after image is basically if you stare at a red light for a prolonged period of time and you stare at something white, you suddenly switch to something white, then you'll see green because red and green are pairs. So hearing comes as sound waves, and then sound waves goes through transduction. So as I said before, transduction is a really important term. It's used to describe neural impulses, stimulus becoming neural impulses, goes to transduction, and it gets sent as neural images to the brain. Remember, amplitude determines loudness and frequency determines pitch. So sound waves is collected in ear and go to your ear canal. So your ear canal, and then you have the eardrum. After it goes to the eardrum, it goes to your tympanic membrane, which is another term for eardrums, and then goes to your oculus. Your oculus have 3 bones attached to it: hammer, anvil, and stirrup, which is right here. These are just different names for it, but it's the same type of bones. And then after that, you go through your oval window. Your oval window gets the sound waves to your cochlea. And then cochlea basically looks, let's see if we can find an image. Cochlea basically looks like this. So it's like this snail shape. And then once you go to the cochlea, your oval starts vibrating. After that, the fluid inside your cochlea starts moving. Cochlea is connected to organ of Corti. So right here. And then organ of Corti is the neurons that activate the hair cells, which gives the neutron information to organs of Corti. Okay, so pitch theory. Pitch theory are three types of theories that explains why you hear things the way you do. So firstly is place theory. So place theory says that the hair cells in the cochlea respond to different frequencies based on where they are in the cochlea. So if they're depend on their location, they react to certain type of frequencies. That is the reason why we hear pitches. And then for frequency theory, it's the idea that lower tones are sensed by the rate at which the cells fire. So however, the rate that it fires is going to be the rate that you hear. Okay, for deafness. So firstly, you have what's called conduction deafness. Conduction deafness is when your hair cells in your cochlea are damaged, and we know that hair cells do not regenerate. So once you have conduction deafness, there's no way to fully recover from it. Okay, touch. We interpret the amount of indentation or temperature change as intensity of touch. High and low concentration, we have high and low concentration of nerve, and they allow us to feel the intensity of the touch. And if it's stimulated sharply, the pain receptors will get the neuron message. And then we have the gate control theory. Gate control theory says that some pain take priority over other pain. So a high pain opens the gate and small pain doesn't enter. That is why when you have an itch, you scratch it. So when you scratch it, you feel the pain of scratching that is of higher priority, and this shuts the gate to the itch so you don't feel itchy. Okay, taste. We taste things, for example, food, because we have papillae, which is at the back. All of your taste buds are very concentrated in your papillae right here. Smell. For smell, first thing is that molecules settle in your mucous membrane, which is at the tip of your nose right here. And then you go to your receptor cells. Your receptor cells sends the molecules to the olfactory bulb, which is right here, and then the olfactory bulb sends it to the brain. That is why you smell things. Okay. Vestibular senses. This is the senses that allows us to balance our body and have like a basic sense of place of where we are. So semicircular canals in your inner ear, which looks like thing right here, it has fluid inside of it. So once you move, your position of your head changes. So once the position of your head changes, the fluid moves. And once the fluid moves, you have a sense of balance, and then your body can adjust to that balance.
Our kinetic sense tells us where our body parts are. It is our body parts are. It comes from the receptors in our muscles and joints, which send information to our brain. This is combined with the visual, so this tells us where we are. And then we look at threshold. So our senses all have limits. Subliminal stimulus would be something that is a stimulus that we cannot register. So for example, there is mosquito noise, which adults cannot hear, and dog whistles, which we can't hear but dogs can, because they are below our hearing threshold. So there's difference threshold, which is how much difference do we need between stimuli in order to tell that they are different. So for example, in terms of pitch, we need a certain change in pitch in order to recognize that one note is different from another. Weber's law Webler's law is pretty much a quantitative explanation of difference threshold. And then we have something called the absolute threshold, which is pretty much what is the minimum amount of stimulus that we can hear. Anything that is below this absolute threshold will be considered a subliminal stimulus. And then we move on to perceptual theory. Perceptual theory is split into many parts. First, we have the signal detection theory. This is where the effects of distractions and interference we experience when we perceive the world. This is how we experience competing stimuli. For example, there is something called the false positive, where we think we perceive a stimulus that is not there. And its opposite, which would be a false negative, which is where we are not perceiving a stimulus that is present. An example of false positive would be if you hear someone call your name when you really don't, and false negative would just be not hearing someone call your name. And then there are two types of processing. Top-down processing is the idea of going from the big picture to the details. This is what tricks us in optical illusions because we generally always see the whole picture and then we see the parts that are actually missing. Our brain tends to fill in the gaps, which might end up tricking us. Top-down processing is always faster, as it is much easier to see the more general view than to see the detail. And then there's bottom-up processing, which is the exact opposite. You go from the details to the big picture. This is evidently much slower, but generally more accurate. So in terms of perception, there's something called Gestalt's rules of perception. The first one is proximity, stating that we have a tendency to group objects that are closer together. For example, here we would see group 1 and group 2. We would see two groups of 4 rather than just a group of 8. Similarity is where we would group objects of similar appearance. For example, if you had this type of shape, you would group the small circles together and then you would group the large circles together. This is something that we do involuntarily. And then there's continuity, where we generally define something to be more similar if it is following some sort of continuous pattern. For example, here we have lines. However, we would define this line as being more connected to itself than with this line, since these two lines are more continuous by themselves. Then there's Gestalt's rule of closure. This is something that's pretty important as it exemplifies Gestalt's idea of how our brain tends to look at things in the big picture. For example, if you had a square that was missing this part, you would still consider it square because your brain would fill in that extra part. And then there's the idea of constancy. It is the ability to maintain a constant perception despite changes. For example, there is size constancy, where we know the size of an object even if we move further or closer, we don't think that the object changes in size. For example, a house. The closer you move, the bigger it looks, the further you move, the smaller it looks. But you always have this idea in your mind that it is still the same house, and even if it looks smaller, it isn't. And then there's the idea of shape. Shape constancy is we know the shape of a mug and we see it from different angles, but the perception is the same. For example, if we see a picture of a door, we always assume that that door is a rectangle, even if it might be drawn in the shape of a rhombus. And then there is brightness constancy or color. This is the idea that if we see we perceive color the same even if changes in light might make it seem different. Then we move on to the idea of perceived motion. First, there is a stroboscopic effect. This is the effect that allows us to see stop motion as actual videos. It's images in a series of still pictures presented at a certain speed. Our mind fills in the gaps and we see it move. Then there's a phi phenomenon, where a series of light bulbs turning on and off at a particular light can make us see a moving light. Try it yourself if you want. And then there's the autokinetic effect. If a spot of light is projected steadily onto the same place on a wall of a dark room, if we stare, it will move. And then we move on to depth cues. So there are two types of depth cues. There is monocular cue, so one eye, and then there are binocular cues, which are both eyes, two eyes. There's only one binocular cue that you will need to know, but monocular cue, there are quite a few. So first, there is a linear perspective, where, for example, we see train tracks. And if we see the lines, we can tell that this is in fact going further away. And then there is the relative size cue, where if something looks smaller than something else, this one, we assume that it is farther. And there is interdisposition cue. If you have one object that is blocking another, the object that is being blocked seems further, and the object that isn't seems closer. Then there's texture gradients, where we assume that objects that are closer to us have more detail than the objects that are farther away. And then there's shadowing. For example, when we draw 3D objects, we usually always have a shadow to demonstrate where the light came. The shadow is also something that shows that there is depth. And then in binocular cue, we only have convergence, or you might know it as retinal disparity. The way that our eyes can tell the depth of something is we tell the difference between our right eye and our left eye. Both of them have a slightly different perspective from each other, and our brain combines these two separate images into one, which has depth. Perceptions. So first there is the effects of culture on perception. There's the very famous Muller-Lyer illusion, where you have two lines where one looks longer than the other, even though they are the same. We have some perceptual sets, which is just this ingrained perception. So a perceptual set is something which determines that we might be more willing to see something as one thing over another. And it is usually ingrained in us from culture. And finally, there is extrasensory perception, or ESP. There are other senses other than the ones that we have listed above, although many people in society and psychologists are skeptical